Hello, everyone. I think we are ready to begin. Welcome to this third edition of our live webinar series, in vivo recording of spinal neurons involved in the generation of pain sensation and application in pain drug discovery. My name is Diane. I am the marketing manager at Neuroservices-Alliance, and I will be moderating today's webinar. This event is the third of a series of live webinars. Each of them features one or several of our neuroscientist experts and showcase their work to show you how Neuroservices Alliance can be your efficient CNS drug discovery partner. We know that your time is valuable, which is why our online events all follow today's format, 20-minute presentation followed by 10-minute question and answer. If you have any question at any time, please don't hesitate to submit them in the question box that is located on the bottom of the GoToWebinar panel. Your question will be answered right after the presentation. I would like to inform you that this session will be recorded and you will receive the recording after the live session. However, I strongly encourage you to stay until the end, as it is the occasion to have our speaker answer your questions live. Before starting, I would like to say a few words about Neuroservices Alliance for those who don't know us. We are a consortium of CNS contract research organizations. Our team is specialized in vitro and in vivo electrophysiological and behavioral studies. Our goal is to deliver endpoints, facilitating decision-making to our clients for their CNS drug discovery programs. We value direct and efficient interactions with our clients, and we rely on a team of 50 PhD scientists. As a speaker, we have Julien Allard with us today. Julien is the founder and CSO of E-Phys. Hello, Julien, and I will let you take the lead from here.
Hi, Diane. Thank you. Very briefly, to present myself, I was originally graduated as a biochemist, and I started to work in preclinical research using anesthetized rodents in 1999, after a PhD in neuroscience involving mostly molecular biology. E-Phys is an heritage from the in vivo electrophysiology team from the pain therapeutic area, which was working at the Pfizer Global Research and Development site in Sandwich in the United Kingdom. The site was closed in 2011. E-Phys was established in 2013 in the School of Medicine of the University Clermont-Auvergne. E-Phys is exclusively focused on in vivo recording in anesthetized rodent for pain drug discovery. I will give you a very brief reminder of the ascending pain pathway in mammals before presenting the recording setup for spinal neurons in anesthetized rodents. I will then present you 4 case studies. Before that, I would like to remind that this very classical drug discovery workflow relies extensively on ex vivo and in vitro assays. The disadvantage of those assays is that it can bring the target into an artificial environment that has little resemblance with the native environment and therefore can lead to inefficient drug design, which is why I strongly advocate the use of in vivo experiments as early as target research and validation, but also throughout the course of lead optimization. At the peripheral level, the axons of the mechanoreceptors that transduce touch-related sensation and the axons of the nociceptors that transduce pain-related sensation travel in the same nerves, and their cell body are mixed together in the DRG. Pain sensation and touch sensation travel within different neural substrates in the spinal cord. The axons of the mechanoreceptors enter the spinal cord through the dorsal horn and travel ipsilaterally within the dorsal colon median lemniscus system. The axons of the nociceptors also enters the spinal cord through dorsal horns, but there they make synaptic contact with neurons. The axons of those neurons decussate and reach the brain within the anterolateral system. The neurons that are activated exclusively by noxious stimuli are thought to be located within the very superficial layers of the spinal cord. Those are called the nociceptive specific neurons. Deeper in the spinal cords, in lamina 3-5, are neurons that also play a role in the generation of pain sensation that receive additional input from mechanoreceptors. The major termination of the anterolateral system differ from one species to another, as well as the distribution of the nociceptive specific and wide dynamic range neuron in the dorsal horn of the spinal cord. In the rats, there are solid anatomical data that have shown that within the anterolateral system, the major pathway is constituted by lamina 1 neurons projecting to the parabrachial area. So, by extension, the scientific community accepts that in rodents, the ascending pain pathway is constituted by lamina 1 spinoparabrachial neurons. There are, however, other pathways that may be involved in the generation of pain sensation, and for example, neurons located in lamina 3-5 of the spinal cord and projecting to the thalamus. The recording of spinal neurons require a laminectomy to access the spinal cord and is performed under deep anesthesia. Adequate physiological parameters are ensured by monitoring and controlling blood pressure, core body temperature, and end-tidal carbon dioxide throughout the experiments. It is also possible to perform a complete blood chemistry analysis at completion of the experiment to ensure that the physiological status of the animal is adequate. There are two ways to search for spinal neurons. One is to apply mechanical stimulations on the paw while moving the electrode up and down in the spinal cord. The other is to use a so-called antidromic stimulating electrode. By placing the antidromic stimulating electrode in the region of interest, for example, the parabrachial area, we can ensure that all the neurons that are recorded from in the spinal cord do project to this region. The type of search depends on the need of the experiment. A wide range of stimuli can be applied on the paw. Brush, Von Frey, or calibrated pinch are used as mechanical stimulations. Thermal stimulations consist in water jet at different temperature. It is also possible to apply electrical stimulation of the receptive field of the spinal neurons. It is also possible to use chemical stimulations, for example, application of capsaicin cream or mustard oil. Those recordings were obtained in anesthetized mice using antidromic search from the parabrachial area as the search stimulus. The first line corresponds to the raw recording, showing the background activity and the action potentials. The line above corresponds to the number of action potentials per 0.1 second beam. In mice, about half of the lamina 1 spinoparabrachial neurons respond exclusively to noxious mechanical and thermal stimulation, such as pinch and water jet at 50 degrees C. Those are the so-called nociceptive specific neurons. There are a few neurons that respond preferentially to water jet at 0 degrees C, called cold neurons. They represent a minor fraction of the population within the lamina 1, but they're interesting because they suggest that there are some modality-specific units within the lamina 1. Deeper in the spinal cord are neurons that respond very consistently to noxious stimulation, such as pinch and water jet at 50 degrees C, but also responds to non-noxious mechanical stimulation, such as brush. Those are the wide dynamic range neurons. In mice, those wide dynamic range neurons are found only in lamina 3-5, whereas cold neurons are found only in lamina 1. However, nociceptive specific neurons can be found in lamina 1 and in lamina 3-5. To carry on with example of recordings, this is the response of a spinal neuron to an electrical stimulation of the receptive field. The electrical stimulation is interesting because it allows to differentiate the action potentials that are generated by C unmyelinated fiber and the action potential that are generated by A myelinated fiber. And this is possible because of the marked difference in conduction velocity of the unmyelinated C fiber and myelinated A fiber that innervates those neurons. There has been recently a number of very spectacular publications that have shown that at least in some pathophysiological experimental model of neuropathic pain, allodynia is caused by the opening of a ventral dorsal circuit in the spinal cord. The opening of this ventral dorsal circuit allows mechanoreceptors that are activated by light touch to activate lamina 1 neurons that are normally only activated by noxious stimulations. The opening of this circuit is due to a phenomenon of so-called disinhibition that can be reproduced acutely by the application of bicuculline and strychnine on the spinal cord. Accordingly, we've shown in anesthetized mice that the application of bicuculline and strychnine on the spinal cord allowed to obtain responses to brush from lamina 1 neurons that were formerly unresponsive to brush. I think this example is important because it shows that in some cases, the pathophysiological dysfunction only occurs at the level of the spinal cord and can only be evidenced using recording of spinal neurons. In this example, recording of mechanoreceptors or nociceptors would be misleading. A very brief word about behavior versus electrophysiology. Behavioral tests have the advantage of relying on a complete integrated response, and they're also quite simple to set up. However, because they include a mechanoreceptive and proprioceptive component in addition to the nociceptive component, they can be misleading. And the best example I can think of is coming from conditional NaV1.7 knockout mice. Those mice do not feel pain. However, their second phase in the formalin test is identical to that of control mice. Electrophysiology can circumvent those confounding effects by focusing on specific pain pathways. I will now present you 4 case studies. The first example corresponds to a pathophysiological characterization. The activity of lamina 1 spinoparabrachial neurons was recorded in mice in control condition and in chronic inflammatory condition. The spontaneous activity of lamina 1 spinoparabrachial neurons was significantly increased in mice with chronic inflammation compared to mice in control condition. Non-noxious and noxious responses to mechanical stimuli were all significantly increased in mice in chronic inflammatory condition compared to control mice. There was also an increase of the responses of lamina 1 spinoparabrachial neurons to autojet at 0 and 24 degrees C in mice with chronic inflammation compared to control mice. The spontaneous activity and the sensitization of lamina 1 neurons is thought to be the substrate for spontaneous pain and mechanical allodynia and hyperalgesia and cold allodynia and hyperalgesia. And this model can be useful to assess the analgesic activity of drug candidate aiming at treating this condition. Spinal neuron recordings can be valuable to evaluate the profile of an analgesic drug candidate. Here, mechanical and thermal responses of lamina 3-5 neurons were recorded in anesthetized mice at baseline and after the systemic injection of either vehicle or the TRPV1 antagonist BCTC. Injection of the TRPV1 antagonist resulted in a complete abolishment of response to warm water jet, a decrease of the response to hot water jet, but it left the mechanical response intact. Spinal neuron recordings can also be useful to determine on which type of nociceptors an analgesic drug acts. By which type of nociceptors, I mean whether those are nociceptors with C unmyelinated fiber or A delta myelinated fiber. Here, the activity of lamina 3-5 neurons was recorded in anesthetized mice, and the response to electrical stimulation of the receptive field was measured. Response was measured before and after a systemic injection of vehicle or a systemic injection of morphine at 3 mg/kg. Responses were similar before and after vehicle injection. The systemic injection of morphine resulted in a significant and marked decrease of the number of C-fiber-related action potentials, showing that morphine specifically and preferentially tackles nociceptive messages compared to mechanoreceptive messages. To finish with, spinal neuron recordings can be used to test tool compounds that could not be used in behavioral experiments. Here, the activity of spinal neurons was recorded in anesthetized mice under artificial ventilation, and this allowed the use of systemic tetrodotoxin. A dose of systemic TTX resulted in the complete abolishment of responses to noxious mechanical, thermal, and electrical stimulations. This experiment could not have been done in behavioral animals because this dose of TTX is just lethal. So to recap, spinal neurons are the first central hub in the ascending pain pathway. Their activity synthesizes the activity of all peripheral inputs, potential changing in the wiring of the spinal cord, and descending modulation from the brain on the spinal network. The recordings can be performed in mice or in rats in control of pathophysiological condition. Drug can be tested acutely. It is also possible to compare the effect of a chronic treatment. And spinal neurons provide a definitive measure of the analgesic efficacy of drug candidate without behavioral bias. So I thank you for your attention and be very happy to answer your questions.
Well, thank you very much, Julien. We already have a few questions, actually. First one, pain models and in fact pain therapeutics have been done by many pharmaceutical companies. How translational is your experimental model to human conditions?
Difficult, more general question. So if we think about morphine, for example, this is a very translational model. Morphine is an analgesic drug in human, and it proved to completely inhibit the nociceptive related activity of spinal neurons. Now, I think the question of translatability is in fact more problematic and will depend on the target.
Okay, and in your opinion, which in vitro and in vivo pain models should be implemented for an improved success in drug discovery?
So I'm an in vivo person, so I regard in vitro as tools to specifically question drug receptor interaction. But for me, the the net analgesic effect can only be measured in vivo. And regarding the models that can be used, again, it depends on the target. I think a very promising target, such as the voltage-gated sodium channel 1.7, can be studied in control mice or rats, well, rather in control mice because of the homology in the extracellular loop between mice and human, which does not exist between rat and human. But a compound that would try to tackle, for example, mechanical allodynia would require a specific pain model. And regarding the allodynia in different models, I recommend the reading of the paper by Genentech, which was the Journal of Neuroscience by Shields, published in 2018, which gives a good idea of some models in which a clear mechanical allodynia is occurring.
Okay. Another question, are the experiments done with multi-channel probes? And if so, how are spikes sorted?
No, only one single electrode is used. And I think that I avoid to do spike sorting because if my take is that if we need to do spike sorting, it's too late. We need a clear signal with a very clear single unit recording with a correct signal to noise ratio. I'm not very happy with the spike sorting method, which is not accurate enough to my eyes.
Okay, thank you. Now, can central spinal cord neurons be recorded from at the same time as peripheral nociceptors?
That would be a tour de force, but physically, no, I think it would be very difficult because that would mean recording from an intact, either intact peripheral fibers or from the DRG. And recording from the DRG directly with a single electrode is something very difficult. So no, I think this would be physically impossible.
Okay, so B, the second part of the question from the same person, do peripheral nociceptors also show responses to non-noxious stimulation following spinal nerve injury in conjunction with central sensitization?
Do peripheral nociceptors
also show responses to non-noxious stimulation following spinal nerve injury in conjunction with central sensitization?
Well, I think in a model of spinal disease, so the answer is no. No, peripheral nociceptors in models of where the dysfunction occurs at the level of the spinal cord do not have changing in their responses. Or that's my take on it.
Okay. Now you say that recording could be performed in pathophysiological conditions. Can you precise how are induced these pathophysiological conditions?
So I've used two types of pathophysiological condition. One was the chronic inflammation induced by a complete fluid adjuvant injection in the hind paw 24 to 72 hours before the measure. So not in the same, it was either 24 hours after or 48 to 72 hours after the injection of complete fluid adjuvant. And so we have quite solid data on that we're working on a paper that was in mice. I also used the chronic constriction injury of the sciatic nerve, and that was in rats. But potentially many, I mean, not any, but most current, for example, a pathophysiological model can be used. I think that it would be quite interesting to study the models of chemotherapy-induced allodynia. That would be one model that could be used, or spare nerve injury as well. So basically, not any, but most models can be used, but they require a deep characterization to ensure that, yes, indeed, the pathophysiological status is reflected in a change in the activity of either lamina 1 or lamina 3/5 neurons. This needs to be documented for any model before doing any pharmacological experiment.
Okay, next question. Oh, wait, were you finished, Julien?
Yes, I am.
Yes. How many units do you get from a typical experiment?
I think, well, no, it's not I think. 2 units from lamina 1 spinoparabrachial neurons in average, and 2 units from lamina 3/5 neurons of a known projection. However, sometimes it's not possible to record from 2 units successively because, for example, if we think about measuring the response to mustard oil, once we've measured the response of first unit, the overall system is changed, and there's not no reason, but there is no interest in finding a second unit. And I have to say that 2 is the average. Sometimes only 1 unit is measured, but on a very successful preparation, up to 3 or even 4 units can be recorded.
Okay, now I'm going to ask a couple questions that were asked before the presentation. How troubling are anesthetics when recording from primary sensory neurons?
I think they are not at all. But, and I can document that. I think volatile anesthetic have a very weak effect on conductions, and so if we record from peripheral nociceptors, volatile anesthetic, urethane, or barbiturates have a very weak effect on their response.
Okay. What are the advantages of recording from DRG versus dorsal horn?
Well, recording from spinal neurons integrates the responses of many DRG neurons, which is kind of an advantage. As I've shown in one slide, there are some examples of where the dysfunction occurs at the level of the spinal cord, and focusing on the peripheral DRG would not allow to measure this dysfunction. And to finish with, when recording from spinal projections neuron, we really know that what we're recording from is the synthetic message that will be sent to the brain. In other words, there is no advantages of recording from DRG neurons if there are changes in their activity that is not transmitted to the brain.
Okay, thank you. And finally, the last question, what kind of anti-epileptic drugs have you evaluated in your model?
I've tested pregabalin in rats with chronic constriction injury of the sciatic nerve. But unfortunately, this was an acute dose, and it was absolutely without effect. And I also used pregabalin in mice with chronic inflammation, again, on an acute basis, and there was no effect of pregabalin.
Okay, well, thank you very much, Julien. I think we don't have no more questions. Thank you everyone for your attention. The webinar has now come to an end, and you will soon receive an email with the full recording of this session. So please do not hesitate to visit neuroservicesalliance.com to have more information about our solutions and to register for our next events. And also, after closing your GoToWebinar window, a very short survey will open up in your browser, and we appreciate your feedback. Thank you, and we look forward to seeing you at our next online events.